All right, so my name is Evan Corundy. I work for the Berks County Conservation District, not too far away from here, over in Leesport. So as our name suggests, I cover all of Berks County. So my main job duty is what you guys are going to all learn about today. I should say one of my main job duties, and that is mosquito-borne disease surveillance. We've also worked into some tick surveillance as of the end of last year as well, so we'll talk about that too. So on a basis of what my job is, I'm on the field collection side of things. So I don't actually get to test for any West Nile virus or anything like that. We send all of our samples that I collect to our laboratory, the DEP laboratory in Harrisburg. So they do PCR testing, which some of you may be familiar with or had saw. Even the lab here has the capability of doing PCR testing to determine the disease prevalence in these mosquitoes and in ticks as well. But I don't get to do too much of that fun stuff. I do some IDs and things like that, which we're going to get to work on a little bit later too. But like I said, I'm more on the field collection side of things. I also do a lot of education and outreach, work with the Envirothon, our conservation leadership schools, a few different other major programs for Berks County students, and also do education and outreach, obviously, on mosquito-borne disease, as well as spotted lanternfly. So I'm sure all of you have heard of spotted lanternfly, and if you haven't, you know, that's a topic for another day. But I also do some spotted lanternfly control work as well. So in a little bit we'll go over the basis of my program. We'll go out then to get to see some of the equipment that I use to do my collections, and then we're going to do some IDing in the classroom with some field scopes. But a little bit of background on how I got to where I am today. I went to school at Blue Mountain High School up in Schuylkill County. From there I took college prep classes to prepare me for East Stroudsburg University, where I got a bachelor's degree in environmental studies and geography. I wasn't sure exactly what I wanted to do. I knew I wanted to do some type of field work. I had always been interested in the environment. I'm a big outdoorsman, big hunter and fisherman, but I couldn't really narrow down what I wanted to do. Didn't know if I wanted to focus on fisheries and take up a fisheries biology courses or wildlife biology. So I decided in the middle ground and take an environmental studies degree or pathway, which kind of covered all the bases. So I took classes with wildlife biology, conservation biology, fisheries, watershed management, you name it. I took a kind of a class in each and dabbled in each. So if you're not set on a specific concentration, you know, maybe you're more into the field biochem type, you know, fields. Even environmental studies majors get to deal with that stuff as well. And you can always go into a higher concentration of that, maybe into a master's program or something along those lines. I don't have my master's program. I knew as part of the field work, you know, bachelor's was really all I needed to achieve what I wanted to. Looking back on it now, it may not have been the best decision I made, but you know, it's always still an option. You get a lot of field experience doing field work, and that builds up your resume as you go. You know, so I can compete with some people that do have their master's programs just with the field experience that I've gained over the years. So that's a little bit of history. I know some of you are pretty young. Some of you probably know what you want to do already. But if you don't let that scare you. I really didn't know what I was going to do. Even in my senior year of high school, I was very undecided. And like I said, it was kind of a last minute decision to not go towards fisheries or wildlife biology and go towards that environmental studies field. And it seems to be working out so far. So we'll get into the program now. Like I said, we'll go through what our mosquito-borne disease control program entails, and then we'll see a little bit of the equipment and get to do some lab. So our official name of our program is Mosquito-Borne Disease Control. That has changed over the years. We used to be the West Nile Virus Control Program because we are specifically looking for West Nile virus, but a few years back there was another disease that was making headlines that was transmitted by mosquitoes, and also travel-related cases were of concern for Pennsylvania, and that was called Zika virus. Anybody remember hearing about Zika virus in the news? I'm sure most of you have. So Zika virus obviously wasn't West Nile, so we were getting things confused, calling it the West Nile virus program, so we moved it towards mosquito-borne disease. Now, at the end of last year, we started doing tick collections as well, doing tick testing. So we've once again changed our name to now the arthropod control program. So I didn't even change it on this slide yet because I'm still stuck in my head with mosquito-borne disease. It took me 3 years to get used to saying that instead of West Nile. Now I have to change it up again. All right, so just to go over here, we're going to talk about the history of West Nile virus, a little bit about the transmission, encephalitis portion, some of my program details, and prevention. All right, so that picture's kind of dark there, but that's a Culex species. That's more specifically a Culex restruens. Culex restruens and Culex pipiens, or the brown house mosquito, are the common species that we are looking for in Pennsylvania. Now, PA has about 62 different species of mosquitoes that we commonly find. There's thousands of mosquitoes out there in the world. Each county, here in Berks, we have about 30, 32 that we commonly collect on an annual basis. Certain counties have different habitats where they will collect other species. Certain counties, towards more of the marshy areas or salt marshes, they obviously have different mosquitoes than areas in the centralized mountains have. But we can usually bank on collecting about 30 different species. But we're kind of focusing on, and we focus all of our field surveillance on these Culex species because they're what we call a bridge vector. They commonly feed on birds, which are carrying the West Nile virus, and can transmit it to us then. So there are a lot of species of mosquitoes that are very specific feeders. They'll only feed on amphibians, they'll only feed on certain mammals, they won't feed on human beings. So therefore we can't get that much disease transmission. Some of these mosquitoes don't even have that capability of carrying this disease. So we're not really too concerned about them. So we focus most of our surveillance on the Culex species of mosquitoes. We're going to talk about one other one then in a little bit here, but that's what our main focus is on. So that is a female Culex there, and she is laying an egg raft on the surface of water, which we'll talk a little bit about later on when we get into our actual traps and how we utilize different collection methods for these Culex species. So a little bit about the history of West Nile virus. So it's commonly found in Africa, Eastern Europe, Asia, and the Middle East. There were several epidemics of West Nile virus in Uganda and Israel. Our first outbreak in the Western Hemisphere was in New York City in '99, and it did not take long to make it to Pennsylvania. First case was found in 2000 in birds, mosquitoes, and horses. 2001, I believe, was the first human case of West Nile virus found. Now it's detected throughout most of the United States. So there's several different strains of West Nile virus, and without getting into some major chemistry of this, ours seems to be closely related to the Middle Eastern strain. So it could have been brought over here in a multitude of different pathways. Whether it was a mosquito brought over, it could have been a sickness brought over and then transmitted through blood transfusion or something like that. More than likely, it could have been the insect itself. All right, so a little bit about the disease transmission. Our primary hosts, especially in Pennsylvania, are corvid species of birds. So you got crows, blue jays, and then also some raptor species as well. But predominantly we're looking into the crow population. So they are, like I said, the primary vector for the disease. A mosquito will feed on a bird that is carrying the West Nile virus, and then that mosquito, the disease takes about 2 to 3 weeks to replicate within its system. And then when it feeds on another species, whether it be human or another bird, it can transmit this disease to that bird. So let's say it's a mosquito that is not carrying West Nile virus feeds on a diseased crow, okay? It will ingest that blood meal from that crow. That West Nile replicates within the system of the crow, and then once it replicates in the system of the mosquito, it has the ability to infect other mammals, humans, crows. So if they go and feed on a bird that is not carrying that host or not carrying that West Nile, they can then infect the bird population. So as the summer goes on and more mosquito populations are building, they're also replicating this virus at a faster rate, which is why we don't find very much West Nile virus in the earlier springtime. But in that August, September time frame, that disease has enough time to replicate within that bird population and expand over the region. So we are what is considered an incidental host, as well as horses. Horses can get several different diseases from mosquito vectors. Like I said, there are only certain mosquitoes that will be considered bridge vectors and feed on humans. But we are basically a dead-end host at that point. So as the crow can be fed upon, and then another mosquito could come along and feed on that crow, replicate that disease even further, that is not possible with humans. So if a human is bitten and contracts West Nile, we cannot be bitten by mosquitoes and replicate that disease. You kind of understand what I mean? We're what we consider a dead-end host, meaning we can be affected by the disease, but we can't then transmit it locally or through a mosquito. It can be transmitted through blood transfusions and direct blood-to-blood contact, but besides that, it is no effect to other mosquitoes. All right, so the incubation period, this is the West Nile encephalitis. This is the actual disease that can affect us. It has an incubation time of 3 to 15 days, so it takes time to replicate within our system. So symptoms of West Nile virus can be none. Usually a healthy host will not exhibit any signs or symptoms of this disease. But it could reach fever or headache, maybe some swollen glands, things of that nature. Some severe infections can result to more severe symptoms. When it reaches encephalitis, which means the swelling of the brain, you can reach high fever, disorientation, coma, and even death. West Nile virus does unfortunately kill several people. It's not anything major in this part of the world, but we did lose 8 people last year in Pennsylvania to West Nile virus. Now, generally, the people that are most affected or affected severely by West Nile virus are either very young children or the elderly because of their compromised immune system. They may have a compromised immune system, so the disease affects them a little differently. Usually it's about 1 out of 5 healthy adults will show any signs or symptoms of West Nile virus. They may think they have the flu, might be flu-like symptoms, and another 1 out of 5 of that small subset will see any type of severe signs or symptoms of West Nile. So it may seem like it's not a majorly big deal, but it is something that we can control and that we can control on more localized levels to hopefully not have any infection or any human infection in the area. So there is no cure for West Nile virus, as well as there is no pre-treatment. You can treat the symptoms, but that is about it. There's no vaccine for West Nile virus as far as humans are concerned. There are vaccines for horses and other equine to prevent the West Nile or Triple E or other, Triple E stands for Eastern Equine Encephalitis, that can affect these horses and equines negatively. All right, so this here is a different species. It's known as Aedes albopictus. This guy is called the Asian tiger mosquito. I'm sure some of you have come in contact with these critters before. They're a very small black and white mosquito with very bright banding of the legs. And you can see the last portion, the last segment of the leg is a white band. That's how we identify the Asian tiger mosquito. So interestingly enough, this is an invasive mosquito. As its name suggests, it came from Asia. They're a container breeder of mosquito. So they will breed in trash, in tires, rain gutters, similar habitat to that of the culex, but the culex also will breed in natural containers, in ruts and tires, or ruts and ponds and puddles maybe, and we'll get into a little bit of their habitat in a future slide here. The Aedes albopictus is known to be a very voracious biter, and it's one of our major nuisance species that has gained a little bit of importance once we heard about Zika virus. A cousin of the albopictus species, the Aedes aegypti, is the primary vector for Zika virus, and it is believed that albopictus, the Asian tiger mosquito, which we have a large population of here in Berks County, has the potential to transmit Zika virus. So we have become increasingly concerned for the Aedes albopictus and controlling their populations as well. So we continue to do that, and we use some different surveillance methods to look for these than the culex species, which we originally talked about. But these also do have potential to carry other diseases as well. Interestingly enough, most mosquito species are crepuscular. They're active during the early morning and the later evening hours, low light conditions. These Asian tiger mosquitoes, they will bite in the middle of the day. It can be high sun, and they're still out feeding. Interestingly enough as well, they're more localized. So if you're getting bit by Asian tiger mosquitoes, more than likely if you walk 100 yards in any direction from where you're getting bit, you can find the habitat from them. They're a very localized mosquito species. Culex, and as well as other species, can travel up to a mile, mile and a half to find food or to feed. Not the case with these guys. So a little difference with the disease transmission of Zika virus comparative to West Nile is that humans are not dead-end hosts. As well as we can transmit this to other mosquitoes, as well as to other human beings, whether it's through sexual transmission, other fluid contact or blood transfusions, as well as mother to an infant. The major concern of Zika virus was a condition known as microcephaly in infants, in unborn infants, and it was of major concern. For the most part, for a healthy human being, Zika virus isn't of concern. You may get some minor signs or symptoms, but the concern of microcephaly was very great in a lot of the areas where Zika virus was run rampant, which is more of a Caribbean area. Berks County, as well as many other areas in the United States, have large Latino populations which commonly travel to the Caribbean area. So there was a lot of travel-related cases being brought into the United States. There was only local transmission or mosquito to human transmission in several states: Louisiana, Texas, and Florida. Every other state, such as New York, Pennsylvania, we did have cases of Zika virus in Pennsylvania, was all travel-related cases, meaning that these individuals had gone to an area, more than likely in the Caribbean, and had been bitten by a mosquito, not saw any sign or symptom until they had come back here. But what interests us with this, unlike West Nile virus, is if there is an individual that had contracted Zika virus and isn't showing any signs or symptoms, so not getting any treatment, they could be out being fed on by mosquitoes. Those mosquitoes can then be carriers of that Zika virus. Remember, we said that in West Nile, humans are dead-end hosts, so we don't have that ability to transfer that to the mosquito. Zika virus is different in that respect as we could. But fortunately, it has kind of slowed down. Our travel-related cases do still continue to come in, but it's on a much lower level. And fortunately, Zika virus hasn't become, a household name anymore. It was pretty large concern in 2016 and 2017, but since then, the concern of it becoming like West Nile virus and embedded in our population has decreased. All right, so to give you guys a little bit of an idea, we talked about the disease replication of West Nile virus ramping up in that August-September timeframe. It's very similar with the Zika virus, but it can be pushed back a little bit because we don't see our peak albopictus or our Asian tiger mosquito numbers until August and September. Usually our peak numbers of QLX are in July and August. Because of increased habitat and other environmental factors. So it takes that a little bit longer for that disease transmission. So if Zika virus were to become an issue, more than likely most of our human cases would be showing up in that September, October, and later time frame. But this graph just shows here on the numbers that we had caught and where that peaks. And the numbers that we have been, or in the years that we have been collecting Asian tiger mosquitoes, the graphs are very similar. So just to throw a whole bunch of numbers up there to explain a little bit about last year. I did a comparison of our 2018 to 2017 year. I was going to put some more numbers up here on what we've collected in 2019, but there's already too many numbers up here to didn't want to confuse it anymore. But 2018 was one of our busiest years and craziest years since the inception of our program. So DEP, the Department of Environmental Protection, created a West Nile virus program in 2001 when our first human case in West Nile was found in Pennsylvania. Since then, we haven't really had too many years like 2018. I'm sure most of you are familiar with the abundant rainfall that we had last year. Mosquitoes breed and lay their eggs in water. And there was just a constant supply of that. So our mosquito populations were through the roof, and our disease replication followed suit with many more mosquitoes. If you continue the disease replication throughout the summer, you're going to end up with a lot more disease within that population. So just to give you guys kind of an example of a comparison, let's just look right here. These numbers are impressive. In 2017, we collected 47,000 specimens, so 47,000 mosquitoes were collected, and our collection time frame runs from May until the end of September. In 2018, we collected 86,000, so we had almost double our positive pools as well in 2018 compared to 2017. In 2017, we had 145 positive mosquito pools or positive hits. That means a specific site went positive. In 2018, it was 243. So these numbers are a little confusing here. So in 2018, we had 243 positives, but 362 pools. So a pool of mosquitoes is what is collected. In PCR testing, what you do is you take the mosquitoes that you collect from that trap, you grind them up to do the testing. But usually it's a mosquito pool of 50 to 100 specimens. If we were pulling traps with 3, 4, 5, 800 mosquitoes in it, we had multiple pools being tested from that initial site. So to coincide with our surveillance, we can't mix pools. So one pool from one site would have to be tested. So in a lot of instances, and the reason why this number is so much higher than the number of actual positives that we collected is because we had many traps last year with hundreds of mosquitoes in it and multiple positive pools from that, meaning that we had many mosquitoes that were going positive in that specific area, which is not a good thing, not a good thing for the human health concern. Just to give you another idea, we're going to talk a little bit about these traps later on when I show them to you. We have grabber traps and we have BG sentinel traps, two different types of traps. I should have pictures of both of them in here. but they are the two collection forms that we that we utilize to collect mosquitoes. There's many other different traps, but these are the two that work best for us and for collecting the species that we are looking for. All right, so our goal is obviously to combat the spread of West Nile virus through many different forms: education, surveillance, and control. Control is a major part of our program. So our West Nile virus season, as I said, was from May until September. We continue to do some sampling into October, and we also do some larval surveillance in the springtime in April to try to knock down those mosquito populations earlier, just to make it easier for us later on in the summer. So education, we do a lot of newsletters, we do a lot of social media outlets, we send notifications and are in constant contact with our municipalities throughout Berks County to notify them what's going on, notify them of our program. If they're receiving any complaints of mosquitoes or areas that may be breeding mosquitoes, they contact us, and then we can take care of that. We have many different educational events too that show our program and showcase what we're doing. And obviously what we're doing here today is demonstrations and presentations to the different camps and different educational groups. So our surveillance. What we do is our adult surveillance. Our larval control is a little bit different. Our larval surveillance and larval control is a little different than our adult surveillance. Larva, we're just looking in different areas, locations for breeding mosquitoes, for those larvae. So we're dipping, what we call a dipper, and I can show that to you later. It's basically like a soup ladle on a longer stick that we can reach into these different areas. So we want to look at common areas that are going to breed these Culex mosquitoes and sometimes Aedes mosquitoes, those Asian tiger mosquitoes as well. So we're dipping a lot of retention basins and detention basins that aren't functioning correctly, that are holding water. We're looking for ruts and puddles. We're looking for artificial containers, tires, areas of larger garbage that can collect water, pools that haven't been opened that are collecting water on the top. A lot of people, when I talk about our program, they ask, why aren't you dipping these wetlands and things of that nature? And there are mosquitoes that breed in wetlands, but for the most part, they're not the species that we're after, and they're a more specific feeder of mosquito, usually an amphibian of some sort, and they're not going to bother us. So we're not very too concerned with wetlands because they have established ecosystems, they have clean water, and they're not really going to breed mosquitoes. So Culex species of mosquitoes, they like organic water because their larvae need to feed on this organic matter in that water. So if you have a wetland that's constantly filtering water, it's usually very clean. When you think of a wetland, you should think of very clean water. So really, these species of mosquitoes do not like that area. They like the tire ruts, they like puddles, they like tires themselves, because that water can gather in there and become stagnant, and there's no predators for them in these ecosystems as well. If you think of it a wetland, you have lots of amphibians, you may have some fish, all of which that are going to eat mosquito larvae and reduce their numbers quite drastically. Interestingly enough, we find that basins that we receive complaints on usually aren't the issue. It's usually something around that basin. It may be a tire rut that's on the outside. We may have a half an acre basin full of water. There may not be any mosquitoes in it, but you could have a tire rut on the outside from mowing in wet conditions, and there's thousands and thousands of mosquito larvae in there. So it's, there's a lot that goes into our job in trying to determine this habitat and trying to reduce their numbers. So that's our initial step in our program. Try to find these problem areas so we can continue to go to those locations throughout the summer and control their population at the source of larvae. For two reasons. One, it's easier to control something that's in a container that can't leave, and two, it's much cheaper. Controlling larvae is by far cheaper than controlling the adults once they hatch. And it's a lot harder to come in contact with adult mosquitoes because they can fly and they don't stay in one spot. It's also, I don't like throwing the word out, safer for the environment, but the pesticides that we use for larval control are generally a bacterial-based insecticide. It's something that, you know, is not harmful to any other aquatic life, maybe black flies and some other non-beneficial insects, but won't affect any macroinvertebrates, won't affect any fish or amphibian species, and won't affect anything that may be eating those mosquito species as well. Generally, our bacterial insecticides that we use are ingested by the mosquitoes or the mosquito larvae, and that is how it kills them. So it is much cheaper, like I said, and a little bit safer as well. So jumping back to that adult surveillance, before we get into adult control, adult surveillance, we have to use a few different types of traps. So this one here is a gravid trap. Okay, so a gravid trap, it's basically a modified toolbox. I'll show you guys, and we'll even have you guys set one up outside of my truck. But the idea is to suck in female mosquitoes. So I know this is a pretty well-educated group, but why do we only want to catch female mosquitoes?
Because they have babies.
They do have babies, right? Right. Now we're looking for the blood of these mosquitoes. So do male mosquitoes take a blood meal? Nope. Male mosquitoes do not feed on us. It's only the females. They require that blood meal in order for more protein to produce their eggs. Male mosquitoes, they'll eat pollen, dust particles, and basically are just here to breed with the females to produce those eggs. They're not breeding, or they're not feeding on humans at all. So we designed traps to specifically collect these female mosquitoes in order to collect and test the blood using that PCR testing in our lab. Just because if we collect a whole bunch of male mosquitoes, it wouldn't really benefit our program too much as far as testing for West Nile. So these gravid traps are designed to specifically catch females. Now we may catch a male mosquito here or there, and once we look at a trap, we'll actually get to see a male mosquito, I'm sure. But the way we do this is by putting water inside a collection chamber that is attracted to mosquitoes. So it's basically just stinky water in a tray that has this modified toolbox on top of it that will suck in the female mosquitoes when they come in to lay their egg masses on the water's surface. And like I said, we'll get to see that. The BG trap, let me see if I got a picture of the BG trap. So here's a BG trap, a little bit different. This is, however, designed to capture mosquitoes that have not taken a blood meal yet. So they are actually, they're what we call a host-seeking trap. So they're looking for something to breed. So we're interested in mosquitoes that are feeding on human beings, so we try to replicate that. Now obviously this trap doesn't look anything like a human being, but it exhibits some additional components that will help trick a mosquito in thinking that it may be a human being. So this cooler up here contains dry ice, which is carbon dioxide, so it mimics our breath, which is one of the attractants that mosquitoes will use to find us. And there's also some human scent lure, which I'll show you guys out of the truck as well. So this human scent lure then will attract those mosquitoes before they also get sucked into a collection chamber here, so that we can sample them.
So when you say human scent, are you just talking like we collect sweat off of a human being?
It's dead skin cells inside of, and it smells like a locker room. It smells like, I'll let you guys smell it. It's not the greatest thing to be dealing with in the hot summer, but it smells like stinky, sweaty feet. Yeah.
A couple of years ago, we borrowed 3 of those from DEV.
Okay.
And so things, just so you guys can, if you're wondering, like, how can I turn this into a project? We got 3 of those BG traps, and a student put them. One was like out in like an agricultural field. One was right by their house, and then one was like in a town, like right in Burgersville or something. And so they wanted to see like what the different mosquitoes like in a field versus in a city. And then they collect the mosquitoes and then identify them, like what we're gonna do later. So even something like that, where if we can set up a couple traps and we can see what different species of mosquitoes are in these different places, that would be an awesome project. So,
Yeah, that would be a cool project. So another thing that we also do is dead bird collection. So we can test dead birds that may have contracted West Nile virus if it's exhibiting some weird behavior. You know, we get a lot of calls about a crow or a blue jay that's sitting in a yard that doesn't appear to have a fear of humans, and you know, it eventually may die. We will go out and we'll collect those birds, those specimens, and test them as well. Again, I don't get to do that fun testing. We send them off to the DEP lab, but that can also help determine if West Nile virus is present in that area. We also do major habitat reduction events. So one thing that we had started when I had taken this job is tire collections. So we know that abandoned tires are a major source of mosquito breeding and a major source of also some unsightly areas. So we figured we'd take care of two issues with one solution, and that's doing free tire collections for Berks County, where residents and municipalities can bring tires that they're not using anymore, that have been abandoned, that are breeding hundreds to thousands of mosquitoes each year, and we can recycle them, and they get recycled for playground equipment and things of that nature, or AstroTurf on fields. So it is actually utilized. We also do some habitat reduction in the form of basins that are not functioning correctly. So most retention basins in a town are designed to hold water for a certain amount of time and then allow it to infiltrate into the ground. Sometimes they get clogged up with sediment and they just continue to hold water for a longer period of time, usually enough to breed mosquitoes and become a major issue that I have to treat on a relatively weekly basis or a monthly basis. And we have corrected those issues or make sure that the correct basins are being installed and implemented in these areas. So that's just a way we can reduce this habitat throughout Berks County.
So with all the rain that we've been having this year and last summer, your job's gotten harder?
Yes, it has. Yes, it has. Last year was very busy. You guys saw some of those numbers with the number of positives that we had. We're getting into the form of control, adult control here. We did the most adult control events last year than we had ever done before, and we would have done double of what we had done if we weren't getting canceled for rain. We can't apply our pesticides in rain, and I was waiting at the truck for many nights, and we'd have to cancel at the last minute because of a storm or weather that was coming through the area. Fortunately, this year it seems like my job's gotten a little bit easier and not so busy because while we're still capturing quite a few mosquitoes, it seems that last year had a major hit on our vector, on those birds. So our host population has decreased, what we believe has decreased a fair number, so that disease is not replicating as much. Matter of fact, we only have pulled one positive so far this year. Last year at this time we had 50 or 60 already. So it does seem like our numbers are being reduced. I didn't put this figure up there, but in May, we had 50% more mosquitoes collected than in any other May in the inception of our program. So we thought, oh boy, we're in for it again. It's gonna, even if our host population is down, we're still gonna have that many mosquitoes. We're gonna get nuisance complaints. Eventually that replication is gonna build up in what hosts are left. But oddly enough, in June, it decreased to 50% less than any month that we have collected in June. So we're not really sure what's going on there because there's plenty of habitat around, but our trap numbers have not indicated an increase in population. It seemed to be kind of decreasing, leveling off, if not decreasing even. And even our larval surveillance, usually we're out doing larval surveillance throughout the week. And we just have not been finding very many breeding locations. So we may be in for a pretty easy year actually as far as disease. Now there are localized areas. There are plenty of areas that are still inundated with mosquitoes. But again, it's not a widespread issue like it was last year. Last year, just everywhere was an issue. It was hard for us to keep up. So we're getting into the control portion of this. So larval, like I said, we treat using the bacterial insecticides. It depends on what type of mosquito, what type of habitat we're using, and that will determine what type of insecticide we use. Like I said, most of them are pretty safe for the environment, but we still do carry a pesticide applicator's license, so I have to take a test in order to allow me to apply these insecticides to the environment. This is our last resort of control. We'd much rather do habitat reduction than actual control. Like larval control isn't that serious, but adult control, if it does make it to that, is a lot more involved, a lot more expensive, and there are a lot more factors that have to be taken into consideration. So once it makes it to adult control, what determines that we do an adult control is thresholds that we create or that we set in place even prior to any season starting. So we have a certain threshold of mosquitoes per trap that are carrying this disease, and this is for a specific location now, that we have to collect before we do any adult control. So how we do our adult control is with a truck-mounted sprayer. So this picture is kind of crummy because we have to do it at night. But we have a truck-mounted sprayer with a ULV machine, which stands for ultra-low volume. I couldn't bring that with today because it takes up a whole bed of my truck. But this machine is basically a pump that sprays this insecticide that comes in contact with the adult mosquitoes, with them flying around, and will knock them down. So we have to apply this at night because the insecticides that we use will also kill honeybees and other beneficial insects. So we do not want to see that affecting our bee population in any way, shape, or form. So we usually start our control right at sundown and go until about midnight. Now there's a lot of other environmental factors that have to play. Obviously, we kind of mentioned rain as one of them. The rain droplets will actually come in contact with our spray pesticide droplets and will knock that out of the air. So we do not spray when there's any type of rain in the forecast. We also can't spray when there's a temperature inversion, which basically won't allow the pesticide to drift as it should. It will keep it grounded and doesn't allow it to drift. Same goes for wind. If we have less than 2 mile per hour wind, our pesticide will not drift enough to get a good enough contact. We're using such a small amount of pesticide that these environmental factors play a huge role. Just to give you guys kind of an idea, we're using a ½ ounce of pesticide per acre. So an acre is a pretty large area, 209 feet by 209 feet, and we're only using a ½ ounce. So it's a very, very fine mist, and our droplet sizes are very, very small to come in contact with these mosquitoes to knock them down. So there's a lot of factors that come into play. So last year we did 9 adult control events. I said that we would have done double that, and I almost guarantee that we probably would have been close to 20 if the weather would have cooperated, but it was almost raining every other day. And because we are applying a pesticide, we have to give public notice 48 hours in advance. It's been reduced to 24 hours in advance now, but last year we had to give public notice 24 hours or 48 hours in advance to any municipality that we were spraying in. So if I was set up to spray on a Tuesday night and it rained, I had to wait another 2 days before I can apply that pesticide because I had to re-announce that to that municipality. So it just kept getting pushed back and pushed back to where we were not getting these controls in. The last day for our spray program is the end of September. We were ready to go, and unfortunately it rained. So there were certain areas that didn't even get tested or sprayed last year just because of weather. We also have to deal with pesticide hypersensitivity. So there are some people that are very sensitive to these types of insecticides and pesticides that we're using. So we can't apply any pesticides within 500 feet of their residence or their place of work, as well as we don't want to spray any of our ULV equipment 500 feet or closer to a exceptional value or high quality stream or waterway. So anywhere there is a water body that's considered exceptional value or high quality, we must stay away from those areas as well. So this really can make it difficult to plant our spray routes because usually we're only getting about 200 to 300 feet of drift. So let's say in a town, for example, we have to go up and down each street corner to make sure that we get a sufficient amount of drift within that habitat to provide the knockdown and control of these mosquitoes. So I kind of showed you guys this slide already. So larval control, we apply these bacterial insecticides. It's usually in a granular form. This bacteria is usually attached to some type of media. It's usually corn cob, crushed corn cob, that we usually will either put out by hand or use one of these backpack sprayers in order to treat an area. I did mention the ULV treatment as well, where we would do them at night. The larval control, that can all be done during the day because, like I said, they have to ingest it and it's only attacking those larval mosquitoes. You can also buy control products yourself, which utilizes the same active ingredient as what we are applying, but it's a non-registered or non-restricted use, meaning anybody from the public can buy them. So we do suggest to people that have mosquito issues in their backyard, maybe they have a little koi pond or something that doesn't have any frogs in it and it breeds mosquitoes, they want to put a mosquito dunk in it. Maybe they have a rain barrel that they can't flush through on a weekly basis or in time before the mosquitoes breed. They can put one of these mosquito dunks, is what we call them, in that to control it. So there is forms of control that even the general public can do on their own property. Prevention is a big thing. So the main way to reduce mosquito populations is to control them from the beginning, and that's reducing their habitat, their breeding habitat. So any standing water in your yard can become a breeding site. So clogged rain gutters, bird baths, window sills, rain barrels, like I had said, pools that aren't opened, wheelbarrows, buckets, flower pots. Anything that can hold water can become a potential breeding site for mosquitoes if it's allowed to remain stagnant and build up that organic matter in it. Usually in the summertime, it takes about 5 to 7 days for this water to become stagnant. I've seen it happen a little bit quicker than that. When it's colder, usually takes about 7 to 10 days. But if you have some type of container holding water, it's a good idea to turn it over every 5 days, and that'll prevent your mosquitoes from breeding. So mosquitoes go through several different life stages. I was going to mention this in the lab, but it kind of ties in with this here. They go through instar stages, meaning growth stages. So they are hatched from the egg, go through 4 instars of larvae. In that 4th instar stage, they stop feeding, so our control doesn't work anymore. Before they pupate, they turn into a pupa, and then they hatch into an adult, or morph into an adult. So we have to control them at those first 3 instar stages. But if you're just dumping water out of a bird bath, even if you see the pupa, which is usually around day 6, 7, you're still killing them because they can't survive without that water. So if you just simply dump that water out, you're controlling those mosquitoes. And then a little bit more prevention. We're always going to have mosquitoes. We're never going to be able to get rid of every mosquito. That's just an impossible task and something that we're not even attempting to do. But reducing this type of habitat in the yard is a major step and a very good first step. When it is dusk and dawn, these mosquito species that are commonly active at this time, wear long sleeves. If it's too hot like it has been lately, long sleeves and long pants aren't really an option. There are products that you can apply. Usually something containing DEET works very well. Some people don't like to apply DEET, so there are certain remedies using peppermint oil and some eucalyptus that are effective. I don't think they're effective as DEET, but I also don't like to apply DEET as well, so I do understand why people try some of these other forms. There's also clothing now that is injected with permethrin, which will help prevent those mosquitoes from biting you, even ticks as well. They don't like that. So that's another option that you can help prevent mosquito bites and tick bites as well. All right, any questions for this board?